Assalamualaikum and greetings. My name is Snail Suryati binti Satimin and I'm from Group 802205A. Today, I will present my final year project proposal. The title of my final year project proposal is The Study of Golden Apple Snail, Pomacia canaliculata population on Paddy Field at Kg.Kulambai, Kota Belud, Sabah. Okay, let me start with the introduction. The research background. Paddy or rice, scientifically known as Oryza sativa, is an essential food source for over half of the world's population. It is grown mostly in Asia, which is significant because the region is responsible for approximately 90% of the world rice output. Rice granaries can be found all over Peninsular Malaysia, Sabah, and Sarawak. Some of the more prominent locations for rice granaries are at Muda Agricultural Development Authority, MADA, Kemubu Agricultural Development Authority, KADA, North Terengganu Integrated Agriculture Development, KETARA, Project Barat Laut Selangor (PLBS), Krian, Seberang Perak, Seberang Perai, Kemasin, Rompin, Kota Belud, and Batang Lupar. Golden apple snail, also known by their scientific name Pomacia canaliculata, are invasive snails belonging to the family Pomacia and are considered to be one of the most damaging pests for paddy cultivation. Some of the golden apple snails manage to make their way into the rice field through the drains. Because of the harm they cause to rice plant when their number gets so high, golden apple snails have changed from ornamental animal to an unpleasant pest. The golden apple snail causes harm to rice plants, beginning in the nursery and continuing for about 4 weeks after planting. When the golden apple snails attack, it cuts the best of the immature rice stalk and killing the entire rice clumps. Next, we move to the problem statement. Golden apple snails have a ravenous hunger for rice plants, particularly the leaves and stems of the plants. The way of consumption causes considerable harm to rice crops, which it in turn leads to a decrease in production and economic losses to the farmers. The snails have the ability to quickly reproduce and infest wide areas, which makes the problem even worse. Golden apple snails are notorious for their high level of adaptability and resilience, which make it difficult to keep them under control or manage them effectively. Additionally, the snail lay eggs in distinct clusters of bright pink or orange above the waterline, which makes detection and removal of the snails difficult. Next, we move to the objective of this research. The only objective for this study is to estimate the population size of golden apple snail in the paddy field. Next, significance of study. This research is about identifying the population and effects of the golden apple snails in paddy field at Kampung Kulambai, Kota Belud, Sabah. The case study will be carried out at one of plot of the paddy fields. The type of data for the case study is the number of golden apple snails population. And next is limitation of study. This study's limitation is the limited resources for this research, such as the article for the golden apple snail population on paddy field in Malaysia, published from 2019 to 2023. Other than that, the quadrat that will be left on the paddy field after sample 1 are taken may be disturbed by uncontrollable organisms such as cow and also other humans. Okay, next we move to the literature review. The first one is paddy cultivation in Malaysia. According to Dorairaj and Govender 2023, in the agricultural sector of Malaysia, rice holds the position of being the third most significant crop, following the prominence of oil palm and rubber. Rice production systems are impacted by diverse biological and climatic conditions, and they comprise rain-fed and irrigated lowlands, uplands, and deep water environment. This system can be found in a variety of geographic locations. The primary source of rice production is derived from the irrigated lowland rice system, which constitute approximately 75% of the worldwide rice output, equivalent to approximately 93 million hectares. Dorai and Govenda 2023. In early 2019, Malaysia's paddy and rice production amounted to 2.9 million metric ton and 1.88 million metric ton, respectively. Additionally, the self-sufficiency level (SSL) was recorded at 72.85%, Ramli 2019. The second one is golden apple snail, Pomacia canaliculata. According to Rahim and Joshi 2019, the unintentional introduction of this invasive snail into Malaysia occurred as early as 1992. This species is challenging has demonstrated remarkable success in infiltrating freshwater ecosystem and exerting significant influence on aquatic habitat. Notably, its impact is most severe in rice fields, where it actively damages the growth and development of paddy stem, resulting in substantial economic implications. According to Rusli 2020, the golden apple snail, widely recognized as a significant pest in the cultivation of flooded rice across tropical and subtropical regions in Asia. A population of golden apple snails infiltrated to rice fields through the drainage system. The species underwent rapid reproduction, resulting in a significant population growth within a short span of time. The third one is capture-recapture sampling method. According to Bukhari 2019, capture-recapture method of sampling are being applied in order to estimate the population size of an unidentified population. The technique of capture-recapture sampling is an often used method for estimating population size. It involves the repeated sampling of individuals within specific population or areas, specifically targeting those that have been previously marked with unique identifier. NextGen 2022. Capture-recapture sampling is a frequently applied technique for estimating the size of a population that is unknown, as it is frequently challenging and unattainable to manually count each individual entity within the population. NextGen 2022. The method often provides a statistically sound approximation of population size, exhibiting greater efficiency and timeless compared to a census in multiple conditions. NextGen 2022. The fourth one is capture-recapture data analysis. According to Bukhari 2019, the Peterson Index method, the Chapman estimate method, and the Schnabel estimate method are the three fundamental approaches that are applied in capture-recapture sampling. The Lincoln-Peterson Index, which is generally accepted as standard method, is typically used as a basic measure in capture-recapture analysis. The first formula is Peterson Index. In this formula, N is for number of population, M is for marked organisms, C is for captured organisms, and R for recaptured organism. The second method is Chapman method. Chapman estimator is called as a modified Peterson Index. And the last method is Schnabel estimate. This method is when the organism marked in the first capture and then released back to the population. After that, a second sample is taken after a relatively short amount of time. The organisms in the second sample that were not previously marked will be marked, and this process will be continued until this final sample. In this formula, MT stands for the individuals that has been marked, CT for individual who have been captured, and RT for the individuals who have been recaptured at a time T. Next, we move to methodology. The experimental site. The location that is chosen to gather information on the population of golden apple snails in paddy field is at Kg.Kulambai, Kota Belud, Sabah. The plot's location is 10 kilometers from Pekan Kota Belud. Total area of the paddy fields is 6 acres, but the data will be collected from one plot of the paddy fields only, with an estimated area of 0.54 acres. The figure below shows the study area through Google Earth. Next is experimental design. The material utilized in this study encompass the tools and equipment used to collect and record the outcome of the golden apple snail population. The study utilized a range of equipment and materials including quadrat, plastic basket, nail polish or correction fluid, tissue, and notebook. The following table present a various material accompanied by their corresponding descriptions. The first one is quadrat, 1 meter by 1 meter. The wood stick and yarn will be connected to be a quadrat on the paddy field with the size of 1 meter by 1 meter. The second one is plastic basket. Together the golden apple snails that will be collected from the paddy plot. The third one is nail polish and correction fluid to mark the snail shell in the first sample. The fourth one is tissue to dry out the wet snail shell. And the last one is notebook to record the data of nails taken from samples. For the data collection, capture-recapture is a sampling method that will be used to estimate the snail population size. To do this, in the first sample, the golden apple snail will be captured from 10 quadrats placed randomly in the study area using hand-pick method. Then will be marked and released back to the population. Then the second sample will be done after 1 week time interval, which will involve recaptures from the same 10 quadrants. The data of marked and unmarked golden apple snail will be recorded. The ratio of marked and unmarked snails will be used to estimate the population size. For the data analysis, the data collection will be collected at Kg.Kulambai, Kota, Belud, Sabah. The method used to do the data collection is use capture-recapture method. The study area of the experimental site is 0.54 acre for the paddy field. In the first sample, the snail will be captured from 10 quadrats placed randomly on the paddy field. Then, the snails will be marked and released back to the population. The second sample will be taken after 1 week time interval. The procedure to estimate the population is using tunable estimate, where the organism in the first capture will be marked and then a second sample is taken over a short period of time. Bukhari et al. 2019. And this is my research flow chart. The first step is preparing the apparatus, which is for the capture-recapture method. Next, data collection. Total sample for this research is 2 samples. Then, do the data analysis to estimate the population using tunable estimate. And last but not least is result and discussion. This is my research Gantt chart. The table refers to the work schedule for this final year project. It starts from April 2023 until February 2024. Next, the expected outcome. The expected outcome from this study, which the study of golden apple snail population in paddy field at Kg.Kulambai, Kota, Belud, is the estimated number of golden apple snail population in one plot of the paddy field. Other than that, from this study, the presence level of golden apple snail in the paddy field can be determined. By knowing the number of golden apple snail population, agricultural agencies in the area can overcome the infestation using suitable integrated pest management. And the last one is my references. This is all of my references for this research. And by that, this is the end of my presentation today. Thank you for spending your time listening to my presentation. If you have any question, feel free to direct message me through WhatsApp. I have left my phone number at the beginning of this presentation. Thank you once more. Have a great day.